Hello and welcome to Bone Up, the podcast all about bones, how we make them, why we break them, and if we fully understand them. I'm David Armstrong.
Hi, and I'm Richie Abel. And over this series, we're going to be exploring osteoporosis, bones, what we know and what we're yet to discover. And we hope you will join us on the journey.
So, for anyone keen to learn more about our infrastructure of calcified collagen, this is Bone Up.
Welcome, listeners, back to Bone Up for the first episode of 2026. Happy New Year. Happy New Year, David. How are you doing?
I'm doing well, thanks, Richie. A bit stormy here today, but it's first episode of the new year, and I think this is the fourth year actually we've been broadcasting. So it's great. The year of the osteoblast, I believe.
Oh, fantastic.
No, I just made that up, but I imagine we probably should designate. We should Bone Up should designate each year to be related to a different cell in the skeleton and see if it gets any traction.
Well, you heard it here first, everybody. 2026, year of the osteoblast. Speaking of osteoblasts obviously are very important in calcifying our collagen matrix. And David, we were talking a little while ago about how, our whole podcast is based around bone health, and obviously calcium is very important. And even in the introduction to the podcast, we talk about how we're going on a mission to find out about the calcified collagen structure. But really, we haven't actually talked that much about calcium so far, which is a bit of a gap because it's so crucially important.
Yeah, it's interesting actually, because as bone experts, we view calcium very much as related to bone, related to fracture risk, related to your bone mineral density. From the body's point of view, if I can use that rather odd expression, the body seems interested really in calcium as much as the concentration of calcium in the bloodstream, and how the concentration of calcium in the bloodstream enables our muscles to work and our nerves to work and other important functions in the body. And really, the body seems to regard the bones merely as a reservoir of calcium. And I think it's a very good time actually to look at how we regulate calcium in general. We focused, as you were saying, very much at the sharp end in the bones, how osteoblasts work, how osteoclasts work, and how we can use drugs to sort of influence that. But it would make good sense now, I think, to look at how the body regulates calcium, and what the body makes of the whole balance between calcium in the bone and calcium in the bloodstream. And we're right in at the deep end here, Richie, because we've got a world expert on the subject.
Listeners, we're very pleased to introduce our guest today. We have Professor Raj Thakker. Raj is Professor of Medicine at the William Harvey Research Institute at the Queen Mary University in London, and Emeritus Professor of Medicine in the Radcliffe Department of Clinical Medicine at the University of Oxford, and Fellow of Somerville College, Oxford. Thank you for joining us today, Raj.
It's lovely to have you, Raj. Thank you for taking the time to speak to us.
Richie and Dave, look, thank you very much for the kind invitation. You're doing fantastic work, and I think everything you're doing really needs to be supported in terms of engaging colleagues and patients as well. And I think I admire you enormously for doing this, and the privilege is all mine to be here. So thank you very much.
Yeah, it's very kind of you.
That is very kind, thank you. It'll help us to keep going. So we've been reading a lot of your work lately, Raj, to prepare for this interview, and I think we picked out a few things today to talk about which our listeners are going to be really interested in. I guess one really important thing is your work helped to show that the body has a kind of calcium thermostat. And listeners, we all know how important calcium is in bone. And when this thermostat is faulty, people can end up with weak bones. What is the thermostat in the body that controls calcium?
Sure. I think you're right to call it a thermostat in the sense that it's a regulator. It doesn't measure heat, so to speak, which is what a thermostat does, but it is a regulator, but it's so it's like a thermostat, it's a regulator. I think before talking about that, let's just take a step back to realize how much calcium there actually is and why it needs to be controlled so exquisitely. Okay, so if you take for what we always call an average person weighing 70 kilograms, that in biological terms, that was always the sort of thing that we used to talk about. So 70 kilograms you're talking about is about sort of 11 stone in the old-fashioned terms, okay? So quite a slim short person. In a 70 kilogram person, the total body calcium you've got is 1 kilogram. That's everything in the bone, everything, okay, is 1 kilogram is calcium, okay? And if you think about it, what's 1 kilogram? Well, actually, that's a bag of sugar that you buy in the supermarket. All right. So all your calcium is in that bag, if you want to look at it that way. What's unique about calcium, it's not in the bag as an amorphous thing, because if it was, we wouldn't be able to be sitting here and talking to each other and walking around doing all the things that we do, but it's organized into this wonderful structure called bone and the skeleton. And if we didn't have that, okay, we wouldn't be able to walk. Our brains would be a slushy mess around the place. So the skeleton really provides a phenomenal structure in the body. And as I say, it's 1 kilogram like that. The thermostat that we're talking about, or the regulator, what it regulates is the amount of calcium there is any time in what we, for the sake of argument, in the blood, shall we say? Okay, it's the extracellular fluid we call it, but it's the blood. So let's look at how much there is. Out of that 1 kilogram, there is barely 1 teaspoonful of calcium circulating in the blood in all of us. To put it into context, about 5 ml. And so the concentration of calcium that we're looking at actually is, in terms of less than 1,000 of a gram. Okay, it's tiny amount. And this tiny amount in the blood is regulated very strictly, because if it drops below a certain level, because calcium has such diverse functions in all of us, if it drops below a certain level, people will start feeling pins and needles in their limbs. They might get very severe cramp in their muscles. And if it gets even more severe, they can have an epileptic seizure, because it's important for the brain. And if it can also affect the functioning of the heart, because it's important for the conduction of the signals in the heart. And you can get rhythm disturbances as well. So that's if it drops too low. If it goes too high, and it's chronically too high, we have to find out where it's coming from. But it can make people feel very ill, mood changes, they get feeling tired, lethargic. If it's chronic, they get kidney stones, kidney failure. And if the calcium's coming from the bones, because of whatever is wrong and it's being leached out of the bones, then you get very fragile bones and you get osteoporosis and fractures. So that, although it's only 1 teaspoonful, if you want to look at it that way, circulating in the entire bloodstream that you've got, okay, it needs to be regulated very exquisitely. And the control mechanisms are many. So first of all, you get calcium in your diet, and you absorb calcium from the gut. That's regulated by hormones. The amount of calcium you have in the blood, that little amount I'm talking about, is regulated by hormones. And then the calcium gets deposited into the bones, which are in an enormous reservoir, as you can imagine. That gets regulated by hormones. And when there is calcium circulating and prevent it going excessively, you lose calcium, you pee it out in your urine, and that's regulated by hormones as well. So how is all this regulated? Well, if you're trying to regulate heat in your house, as you very rightly say, a thermostat, you have a thermostat. It senses what the heat, how hot it is, and you either turn the boiler on or off. Okay. Same thing. We've got what's called a calcium stat, if you want to call it that, but actually we call it a calcium sensing receptor. It's a receptor because it receives signals, and it's calcium sensing. Okay, and many cells in the body have this, but particularly the parathyroid glands, which are situated in the neck, the kidneys, the bones have it as well, and the pancreas has it. But it's spread. It's widely expressed in the body. So the calcium sensing receptor detects the amount of calcium there is circulating in the blood and in other tissues. And if there is too much calcium, it'll switch off the production of in the parathyroid of parathyroid glands. If there's too much calcium, it'll enhance calcium excretion by the kidney. So it does everything to really try and keep that calcium at the right level, in that exquisitely right level there is. So that is what the calcium sensing receptor is. And because it's a receptor, it's on the cell membrane and it does this work, you can also target it with drugs for when people have particular disorders. So I hope that sort of helps to explain the nature of calcium, the importance of calcium, why regulating it in blood, because it's a tiny amount that's circulating there, but that balance needs to be exquisitely controlled, and there are quite a number of hormones that help us to control that.
That's a really wonderful explanation. Thank you very much. We know then that the thermostats in our homes go wrong sometimes and the boiler won't come on. What happens in the body? Do you ever get faults in the calcium thermostat?
Absolutely. So in this receptor, the calcium sensing receptor as we call it, normally it works, which is good news, thank goodness, and it works very well. It can go faulty. And the reason why it can often go, in the majority of instances that we know of, that it can go faulty is because there is a change in the DNA that instructs the body to make that receptor. Now we've all heard about DNA, and DNA in essence is it's like a book. There's an alphabet and the words there. The amazing thing about the DNA book is it only has 4 letters in it: A, T, C, and G, and everything is made up out of that. So those are the instructions. So sometimes, if, for example, it's meant to be a C there, and it isn't a C, but it's a G, that means you make a different type of protein or a different type of block for that protein, an amino acid, and that can alter the function of things. It's that sensitive. Now, so we call these mutations because that's what the genetic mutations. So if there are genetic mutations, it can make the calcium sensing receptor faulty. It can make it faulty in one of 2 ways. It can either cause what we call a loss of function, i.e. it's not working or it's impaired in its function. And if you've got that, then the patients will have a high calcium in their blood and they'll get problems with that. Like all these things, there's always an opposite to this. Instead of loss of function, if you get a gain of function, in other words, it starts to work pretty more effectively than it should do, shall I say? Okay, yeah. Then you get a low calcium in the thing, and you get consequences of that. All the ones that I mentioned earlier. So when it goes faulty, you can either get loss of function or a gain of function, and we need to recognize what that is, and of course, then take appropriate actions to treat that. There is one other thing that can happen, and by the way, because these are genetic disorders, they not only affect adults, actually, they affect children. And often in children, it's missed because one of the things pediatricians or the doctors who look after have to do, children can't tell you what's wrong with them. They just don't feel right. And you've got to be really smart to pick all these things up and watch and look out for all the subtle signs. And it's important to recognize that these things can affect children. So that's one thing. So the other thing that happens in children is that we will all inherit a set of genes from our dad and a set of genes from our mother. And usually these mutations will occur either on what we call the paternal, i.e. the set of a gene from dad, or maternal, the gene from mum. That's pretty simplistic because they can arise de novo, as we call it. Anyway, but it's only one gene that is faulty, not the other one. Okay, and that's what still causes the disease, and there are reasons for that. Sometimes, some people unfortunately get a mutation involving both genes. And if that happens, you get a very severe disease. It's called neonatal severe primary hyperparathyroidism. The parathyroids get much bigger as a result of this. The calcium is way high, and it's being leached out of the bones like nobody's business. And these poor children, even when they're just barely a few weeks old, 2, 3, 4 weeks old, will have severe dehydration because they'll be losing a lot of water from the kidneys because of the calcium being very high. They will also be unwell. They will get fractures, and not only will they get fractures, but if it involves their ribs and things, they get respiratory distress and they die. It's that severe. And the correct treatment for that used to be, still is surgery to take out the parathyroids so that the calcium comes down again. And children do very well, but you need to get to a very good surgeon because parathyroid glands are very difficult to find, and in children they're even more difficult to find. Because it's not. And you talked about operating on a baby that's barely a month or few months old. So what we do now is we use a drug that targets this calcium sensing receptor to lower the calcium, to buy us time, so the baby, instead of having to operate on the baby when the baby's a month or two old, we can buy some time until the baby is 6 months or even a bit older, till 1 year old, and that makes the operation a lot easier. So, knowing about this calcium sensing receptor, the so-called that thermostat, is very important because it's already got practical effects in looking after people who are not well. So I talked about the receptor being faulty because of something wrong with the genes, if you would look at it that way. And the calcium sensing receptor is one, or it could be something in its pathway, which we've identified other genes that get there. The other way it can occur is because for various reasons in that person, antibodies have arisen to the receptor. And we find this happens quite often in a lot of disorders, which are immunologically based, inappropriate production of antibody. In endocrinology, we find this affects the thyroid, for example, autoimmune thyroid disease. And you can get what we call autoimmune antibodies targeting this receptor. And again, they can cause a loss of function of the receptor impairment or a gain. And it depends on what antibody you've got, okay? And there are colleagues with it. So because we're talking about mutations, sometimes you feel, well, if it's not in the family, it can't be a gene. Well, it can be a gene because it's a de novo mutation. It's arisen in that person, but not in the parents, okay? So it's one way. But the other thing to bear in mind is that it could be because of the occurrence of autoantibodies against the thermostat. So, you need to be pretty aware of that so that you can actually approach this in the correct way. So I hope that explained about how these things go faulty.
Yeah. Could I clarify just for our listeners, when you say you get loss of function of the calcium sensory receptor, does that cause high calcium in the blood or low calcium in the blood, or when you get gain of function? Because obviously these are sensors and there's often a positive or negative feedback. So which way does that go?
Loss of function gives rise to a high calcium, and gain of function gives rise to a low calcium.
Okay, so loss of function causes high calcium in the bloodstream, but from the patient's point of view, that usually means the calcium is leaching out of the bones, so the bones become weaker. So loss of function causes weak bones. Gain of function, well, it doesn't really cause strong bones, but it causes low calcium in the blood.
Yes, absolutely, yeah.
It's interesting because over the years with patients, they will get blood results back, and particularly now with so many patients having apps on their phone that they get sent their blood results. and they'll come in to me and say things, look, my calcium's 2.5 this month and it was 2.3 last month, so I've got more calcium, this must be good for my bones. And often we end up explaining, the calcium day to day in the blood's not that important in terms of how strong or weak your bones are. But actually what you're saying from this perhaps is that we should be paying more attention to serum calcium or adjusted serum calcium over the time. Perhaps those patients whose calcium is 2.55 or something towards the higher end of normal, does that mean they have different settings of their calcium sensory receptor from those other group who are down to 2.2 for most of their lives? Should we be looking at that more?
Those are very important points, David, and I just want to clarify, I think, okay, so you're right. When you've got a loss of function or impairment of the calcium sensitive receptor, the calcium does go up, right? Okay.
Mm-hmm.
But those patients very often do not have osteoporosis and fractures, because what has happened is the whole system's got reset at a high point. Okay.
Yeah.
So I wouldn't want people to go with the impression that if you've got this problem, that you've got to get osteoporosis. Okay. And one of the things is that this differs because it's really a body adjustment issue. Okay. So if you've got a person who, say, has a calcium of 2.6, which is just above the upper limit of normal, and this is because of the parathyroids being overactive and acting on bone to release calcium, and it's chronic, then that person gets fragile bones and osteoporosis because it's long term and the parathyroid hormone is acting on the bone. What happens in this impairment aspect of the receptor, right, is the calcium is high, but the PTH is not high. The parathyroid hormone is not high. It's in the normal range. So it's not high PTH doing the damage and leaching the bones out. Okay. So you need to sort of have a perspective on that. Okay. In that way. What was the other question? Sorry. So that was one question you had. The other the patients. Yeah. So I think if somebody's got a calcium of 2.55, I think the key thing, as we always do in endocrinology, you don't measure just one thing. Mm-hmm. You measure because the whole thing is working on to maintain an equilibrium. You measure the other things, so we should be parathyroid hormone, vitamin D, that kind of thing, to make sure that all of that is normal and in check, and adjusted accordingly. It's like a lot of these things, people sometimes sort of say, I measured the insulin, doctor, it's high. I always thought, well, why did you measure the insulin? If you just do it randomly, as well as I do, after we've had some food, our insulins will be high. That's a normal thing, okay? But if they tell me the glucose is low and the insulin is high, that tells me that's wrong.
Yeah.
So all in endocrinology, what we do is we always have to challenge the system to see if we can disturb the equilibrium to unmask the disease or unmask the faultiness of the hormone or the gene. So I think you raise an important point, but what I would say is I think for those people, you need to measure and keep an eye on their parathyroid hormone, things that help with that.
It's the effect it has on the parathyroid hormone, really, that is the important thing. But
absolutely.
Yeah, and it's just when you were talking about obviously quite significant genetic problems that cause major loss of function or gain of function on the calcium septing receptor, presumably there's more subtle changes over, is there a broad range of sort of phenotype of calcium sensing receptors throughout the population that again, we may be seeing people with various different types, but it doesn't have a very strong clinical, effect on the patient?
No, I think that's right, and these are still early days. So what's happened up till now, and is continuing to happen, is that there's what I would like to call ascertainment bias. So when you and I see our patients in the clinic, it's already been selected and they've got a disease. Okay, so we then go and find that there's something wrong with the calcium sensing receptor and say this is what it is. What we haven't done is to go into the population and say what are the variants here and are they causing anything? Okay, so we have started to do that in various different ways, and this is still work in progress. One of the works we did a while ago was with the team in the United States with Geisinger and with Regeneron, because they've got cohorts of all their patients there who've got on electronic database and sequences. I think there was about 250,000. Okay. So we're beginning to look at the variants there, as we call them, and see if we can match them up with what might be going on. And that's good. There are limitations to this, not on the sequencing side, that's very good, but it's how you're reporting those diseases. It's on ICD codes, and with the best will in the world, sometimes you don't code them correctly, or rather, that can happen, but often you miss things. As we were saying earlier on, you need the clinician to be able to say, this is what the diagnosis is, and get it correct, okay? So there are limitations there. The other one we're looking at also is with the UK Biobank, because that's got some very important information. And I think what you're saying is absolutely correct, that you're going to get people with variants that are so subtle that they're not having any effect. But again, there might be various things happening here. Somebody could have a variant, it's not doing much to the calcium, but it might be they're deficient in vitamin D, either from dietary means. So it could be masking that tendency to high calcium. So if you replace the vitamin D, they'll become hypercalcemic, possibly. Okay, so I think there are all those things that we still need to do, and they are in motion, shall I say. Science always moves slowly sometimes, but those are important questions that we need to try and identify. Mm-hmm. The other thing we found is that the calcium sensing receptor is on the cell surface. It's sensing calcium. Then it sends signals into the cell. So what we've found is that there are molecules inside the cell that are a part of this pathway, that when they have those mutations, they can cause alterations in calcium as well. Some of them are very subtle. They don't affect the calcium so much because there's redundancy possibly in the system. Others can cause more severe problems in calcium as well. So that's the other thing that might be going on in terms of influencing whether the calcium is going up because of a mutation or not.
Yes, you made sense there of my very vague question. You've organised it, and you made my question sound more intelligent by producing some very well organised answers. But yeah, I mean, I sometimes wonder if we come back in 20 or 30 years, will we be testing these things more routinely? Will we be diagnosing osteoporosis and testing all sorts of genetic background to vitamin D receptor, calcium sensing receptor, and producing much more tailored treatment rather than just handing out 70 milligrams of alendronic acid to everyone, But
right. So one of the things that's happening right now, and this is a project we're really actively pursuing, and I hope we can get an answer soon. We have asked this question: when a doctor sends a test off with the patient now, we often get the result coming back because of, quite rightly, we have to be very strict about the criteria we're using to diagnose a mutation in an individual, right? Okay, and these, so if the change that's been identified with the DNA sequencing, and it doesn't meet these criteria for saying this is a mutation causing the disease, it is classified as a variant of uncertain significance. And there are probably more of those now than there ever were before, and there are probably more of those now than have been identified as mutations, not just in the calcium, but other diseases as well, okay? And they really are a major challenge for the medical profession and also our patients, because our patients want a diagnosis. They've come to see us. You're being smart, you think, and you're sending off for a genetic test, and you've got the result, and then you're sitting there with the patient and you're saying, well, actually, this is uncertain, I don't know what to do. Nobody wants that.
Yeah, why it doesn't show you whether that's just checking, someone's sodium level or doing a genetic test. Unless you know what to do with the answer, you shouldn't be. It's what you teach medical students, or you teach your foundation doctor. Don't send a test unless you know how to deal with the result.
No, the problem here is you tested, you sent the test off for a good reason.
Yeah.
You've got an answer, because this is a real conundrum of this variant of uncertain significance. Okay, yeah. Now, one way we're doing this, okay, is we have got a project where we're making, and we're trying to prove this in this one, and some other people have done it in some other one. We're making every single possible change that could arise, and we're using our functional assays to produce a database that say yes, this is, or no, this isn't. Okay, all right, and that's not an easy task. But then, we didn't do these things because they were easy. We tackled them because they're difficult. Okay, we need an answer, and we're really making good progress. But alas, one of the things with everything that happens right now is funding is an issue. We're trying to just get the funding from somewhere to finish this project. We're nearly there with it. Okay, and what I would hope, David, is that our ambition in this is that there are about 30,000 variants that could potentially arise. So you, clinician, could go along and say, here's a database. What's my patient got? This is it. Okay, yeah. Here's a database. They've done the tests on it. Yes, this can be classified as a loss of function. I know what to do. Yes, this can be classified as a gain of function. I know what to do. Yes, this actually has been classified as not doing anything serious. So I can shake the hands of the patient and say, I don't think we need to worry about this one. If you've got symptoms, we need to think of somewhere else, okay? And you're right, in 20, 30 years, well, I hope sooner, certainly with this project, we just need to get this into place and a bit of money to finish it, really. And that we should have an answer on this one anyway, I hope within a year or some other ones are going to be, at the moment, like all these things, making the first few of anything is always a challenge. Once you get it up and running, so we're using this as the first gene because we've got very good, what we call readouts. The thing is, if you're making changes, you need to make sure that what you're measuring at the other end is measurable and real.
Mm-hmm.
Okay, so we selected the calcium sensing receptor because we've got very good readouts and we've got good assays to tell us about that. Once we get the technology or the methodology of making all these things, okay, then we can move to another one where hopefully making all those changes will be easier, because we know what we're doing, but we need to make sure we've got some assays on that. And this is a real challenge right now that we have. We've got the genetic side of things, but it's a question of what's the correct interpretation and how do we use that information for our patients. And that's what we want to try and enhance and give people good evidence to be able to do that with.
Raj, aside from being a clinician and a world-leading researcher, you still manage to find time for lots of other things as well. And you've been very active in the bone community and supporting the bone community. You are editor-in-chief of JBMR, and this is the 40th anniversary year. And listeners, this is really the most important journal in the bone world where everybody wants to share their research. What is the single most important thing that you would like the journal to be able to do to move the bone field forward?
Well, JBMR is a scientific and clinical journal. So we publish discoveries, developments in the field, okay? And I've got a plan with my colleagues this year, and it's already happening. So if I can, let me tell you about this, okay? So what I wanted to do is, one, reflect on our history. So what are the major papers? What advances did they lead to? in the last 40 years. Now, the thing about this is looking back is one thing, but the great thing about history is you can learn from it to look in the future. So I want to try and achieve several things. To say, look, what do these ideas do? And when we publish them, for example, JBMR published some of the first results on DEXAs scans, which we use nowadays to pick up low bone mineral density, and it also related to that about how we can use X-rays to identify the people who are at risk of fractures and things. Okay, all those trials and studies were published in JBMR. We take them for granted today. Not just that, but lots of other treatments, trials, all of those are there, which we take for granted today. And I think the thing to do is just reflect to say what was published in those. I'm really fortunate with my previous editors-in-chief. I spoke to them and said, look, what I'd like you to do is, in your 5 years when you were editor-in-chief, pick out your top 5, your top 6, your top 10. Tell us why that was there. And this is important, not just for people to reflect on, but educationally it's important because people can go back and say, that's where that arose. That's how that occurred. And now, also, when you go back and search these things, say, there was a gap there, and there was a gap there. We need to go back and look at this again. So I think the most important thing is that is already happening. Please go to JBMR, first issue, January. We've already got the first article in there. And there's going to be an article like this from each one of the 7 editors-in-chief from January to July. Okay, and we've got a host of other events happening as well. So if you want to come to the SBMR meeting, you'll also learn more about these things. It's happening in October in Boston. Lovely city, great meeting. You'll see the vibrancy and the energy that's in the community there. So do come. So going back to your point here, why I wanted to do this is, as I say, is to highlight A, the achievements, but B, to inspire the younger generation to say, this is how these guys did it. Actually, it was impossible then. They took a risk. They worked hard, thought it through, and delivered it. I can do this today with my things. And so it really is a learning exercise that will probably I hope it'll definitely make the colleagues who made those important discoveries comfortable and warm to say this is it. But more importantly than that, we go to the future. We use this to say to the young investigators now, to say, look, this is what they were able to achieve with these tools. Look what you've got now. How much further can? So let me give you one example of this. And David will know about it. It led to the discovery of a new hormone or a new molecule. Okay, we know that patients who unfortunately get cancers and they metastasize will get a high calcium and be very ill from that. Now, those cancers might arise because of 2 ways. One is the cancer is spread to the bone, and that's causing the high calcium because it's leaching it from there. Some patients get a high calcium and they don't have any metastases to the bone. So the question is, what's happening there? And a very famous American physician in our field called Fuller Albright recognized this back in the, I might get the dates wrong, forgive me, back in the 40s and the 50s, okay? And said, I think this cancer there is producing a hormone that's leaching this out. And the hormone we know that's doing it is parathyroid hormone. They did the assays and it wasn't parathyroid hormone. So he said, I think this is a parathyroid-like hormone that's causing this, okay? And the condition was called the humoral hypercalcemia of malignancy, humoral, i.e. due to hormone. Well, it took a lot of the revolutions that we've had in developments in molecular biology to come forward to this, and it took absolutely inspirational leaders like Jack Martin from Australia and his team, and also Arthur Broadhurst and his team from Yale, and lots of other colleagues to do the fundamental work and identify that, yes, this is correct. And all that was published around the around the 80s, so 40 years later. Okay, and guess what? It is a hormone. It's called parathyroid hormone-related peptide now. And so, here's the thing. Beautiful clinical observation. That's what doctors do. They're clinicians, they observe, and they try and figure out what's going on. We may not have the answer today, but the next generation will come, hopefully be inspired by this, and make those discoveries again that will help to benefit our patients and our society. David, I think I'm not sure if I hope I got the dates correct roughly.
Well, Fuller Albright, I know sort of 1946, wasn't it? He published some of his big work on sort of HRT and bone turnover. Because I have a bit of an interest in medical history, which you probably didn't know, but there we are. So I think the dates are entirely correct for Fuller Albright. I think it's probably late 40s, very early 50s. Richie's just nodding an agreement here, so he's not going to disagree with us live on air. But he'll do this, Raj. He'll cut out and edit in himself if he's getting something just right, So,
but no, seriously. So going back to your question, Richie, I think the idea of this is to review many of those aspects, okay, and to actually inspire the next generation, to to pick up the torch and actually light it even brighter than we did.
I totally agree. That's absolutely critical. It's important for any field to try and attract the best and brightest people to join them, to pick up the baton and carry it forward. And I think telling interesting and exciting stories about how research in our field has helped to improve people's lives, how it's translated into new clinical healthcare, is absolutely wonderful. And hopefully we'll bring those people in.
And something else occurred to me from what you were saying. Sometimes the really important papers aren't the one that appear to be really important at the time. And some researcher has maybe poured his time and energy, or her time and energy and PhD, into a paper, produced it, it's published, and it lies there. And it's maybe only 20 years later someone comes along and says, that was the key, but it wasn't apparent at the time.
David, read JBMR. Read it all the time. Don't get me wrong, okay? But certainly, please look at this month's issue.
I haven't seen this month's issue, I must admit, but yeah, I'm looking forward to it now.
Look in there, and then we pick these papers exactly for what you're saying. That you look back and you realise that was a seminal paper, or really a critical paper in the field, and really advanced it. Okay, it's been a wonderful, shall I say, endeavor to work with my previous editors-in-chief, the previous ones, and they've been absolutely wonderful, and getting together, discussing these, and why they were significant. Sometimes it was even like there's the paper in this month where Michael Parfitt and colleagues actually, the people were not quite using definite terms. Terms that need to be defined. What do you mean? And that paper has been one of the most highly cited papers, because it was actually, setting the scene on how we define things. Very important.
I cited Parfitt's papers in my PhD.
Did you?
And used his definitions of trabecular bone and the structure of trabecular bone.
Richie, that paper is we've got it in our first article. It's there.
Oh wow!
And I think it's probably. I need to check this. Actually, you check it before this. But it's the very first commentary. It's the very first paper we cite, and then write the commentary on that. So no, I think journals are great, and I think we need to use journals not just to report things, which we discovered, but have an eye to the future to say how are we going to inspire the next generation. Because I think the only reason why, well, I think we are the most successful species.
It depends how you define that again, you see, according to what you said. But
that's why I'm being a little bit cautious. I think we're the most successful species. And the reason why I think we are is that we teach our children how to advance in the future. And it's passing that knowledge on to the next generation that actually, I think, makes us pretty unique.
Yeah. We talked about the history of medicine. It's in the Hippocratic Oath that a doctor promises to teach the next generation of doctors. It's in there as one of the fundamental things of being a doctor.
Well, and I think that's why you and I enjoy medicine so much.
Well, exactly.
So this might bring us neatly on to the last question today. And Raj, I was wondering, what are the guiding principles that have led you through your career in clinical medicine, research science, and leadership?
Well, I became a doctor because I was interested in science, and I was interested in biology and in people, and medicine seemed the right career, if you want to look at it in that way. I think there are many facets to being a doctor. Central to everything I do, there's always a patient. Even if it's looking at the molecular stuff, okay, you can track it back to the patient because everything we do starts off in the clinic. Patients got something that's not right, okay, and trying to answer that is where it all starts off. So I was really lucky. I was working at the Middlesex Hospital as a registrar. I did my clinical training there. We were always taught to think about things. And I was an undergraduate at Cambridge where we talked about lots of things and taught to think critically about things as well. So it all came together. And at the Middlesex, which was the major referral hospital, we saw patients that had been seen elsewhere, but people hadn't got the answers for them. And one of my mentors was brilliant. He said, you will always come across this now. Your colleagues have tried to come up with the answer and they haven't. So the first message is go back to the patient, take a very detailed history and examination, because somebody's missed something. There's a clue there that somebody has unfortunately missed, and you have to find that clue. So, as I say, it always starts off with the patient. If you still can't get the answer, the reason why I came to research is I couldn't provide the answers. And I thought, well, actually, there's only one thing to do to get these answers. That's to go into research. So that's what drove me into research. And in those days, what I was getting interested in was to try and identify the mechanism, because that took me back to my training when I was an undergraduate at Cambridge. And I went to a really interesting lecture by a professor of medicine from Manchester. His name was Stanbury, and he gave a lecture. Actually, David, you might remember this. It was what was known as the Bone and Tooth Society in those days, but now the British Bone and Mineral Society, etc.
Yeah.
And it was entitled "Stuck at the Cell Membrane." You think, what does that mean? But actually, what he was getting at is that we knew a lot about the physiology impinging on those cells, but we hadn't got inside the cell, and we're stuck at the membrane. And that struck a real note in me, actually, to say, yeah, no, I've studied physiology at Cambridge and things, and I'm looking at patients, but we can't get the answers by just physiology. We're stuck at the cell membrane. So I was wondering on how we get inside of there. And I was getting interested in multiple endocrine neoplasia because I'd seen quite a lot of patients with that, and we didn't have answers for that as to what caused it. And it's a devastating disease, really. And then David Weatherall, who was an Nuffield professor of medicine in Oxford in those days and subsequently became research, he came to give the Christmas lecture. This is what I'm always saying to students. Go to the lectures outside your field. You you never know what neurons they'll fire and what synapses will connect to those to generate the next set of ideas. So, it's on your field, it doesn't matter. It's a question of, how you start thinking about things, what inspiration you might get from these. So I'm sitting in this lecture, the Christmas lecture given by David Weatherall, and he's talking about the new molecular genetics. This is about DNA, how you get inside the cell, how you study genes and things. And I thought, that's the way to get in. And try and I was trained to be an endocrinologist. I say I can, this is what we need to do in endocrinology. And then the question is, how do you do that? So you got to go and talk to people, mentors. And so I spoke to the consultant I was working with, Jeffrey Reardon, and to the professor of biochemistry who'd invited David Weatherall. And you can see what's happening here now. They were beginning to help me join the dots to get an MRC clinical training fellowship. I was lucky to get that and being supported by the MRC. And I started to retrain because, believe you me, a lot of the stuff that was happening I hadn't even heard of because it wasn't invented when I was an undergraduate. So and this is the great thing we have in Britain, that we have these opportunities for clinical training fellowships for clinicians to go and retrain in an area that they can then bring to the science. And I'm not the only one who's benefited. All you look around. So that's how it all started, really, and that's what inspires me. It's knowing that you're trying to do something that is actually going to help somebody in this world. And at the end of the day, we're actually at stages sometimes where sometimes it only helps one person. Doesn't matter. If it helps a lot, that's good, but never lose focus about why you're doing this work. It's interesting, it's fun. For me, it's got to come back to the patient.
I think that's a wonderful message for any budding scientists out there. Thank you so much for your time today.
Excellent. Well, look, thank you for your time, Raj, today. It's been absolutely super speaking to you. Thank you very much indeed.
Okay, lovely, great, smashing. See you then. Okay. Thank you very much. Bye then. Bye.
So, Richie, that was very interesting. Raj certainly covered a lot of distance talking about calcium sensing receptor, and really covered a lot of other things as well. I'm going to have to go in and digest that. Just as a sort of a first blush, what would you sort of take away from what we discussed with Raj?
I suppose the first thing I would say is that I really loved his way of putting calcium into perspective. I thought that was a really wonderful example of scientific communication, which I'm going to steal and claim as my own. Second, probably that's where the calcium thermostat idea really clicked for me. The body has a sensor, it's a calcium sensing receptor. It's constantly checking the levels and adjusting the levels. And he explained how that was done by the hormones and the kidneys as well as the skeleton. And I thought it was very interesting where he described the receptor sitting on a cell surface, not just from sort of an interesting biology fact point of view, but he explained that that means it's something that we can actually target with medicines in the right patients. And I thought, again, this is another really good example of clear communication. He's explaining essentially how the anatomy and physiology of the cell is important because we can target it with medicines. And I thought he explained that really well. And I generally feel that molecular physiology is about the hardest bit of biology to explain. The third big takeaway from me was Raj talking about genetics. The two of you kind of had a discussion about this, how doctors can do DNA tests much more easily, but it's actually quite hard to interpret the findings of those genetic tests. And I think Raj said, quite often they get back results and the first view is, well, we're not sure what this means. That's a real challenge for doctors and patients. And Raj explained that the solution to this is going to be about building better evidence, basically testing DNA changes in the lab. So then we can try and work out clear answers to what these tests can tell us and how we might be able to use these tests to improve patient outcomes. And for me as a scientist, that's really, really useful because it helps me to target my research towards something useful. What were your takeaways?
Yeah, I mean, just coming away from talking to Raj, I suppose he focused very much on calcium initially when he was talking calcium in the bloodstream and how the thermostat is set in terms of how much calcium, the concentration of calcium in the bloodstream, and how the body actually doesn't really care that much, if I can put it like that, how dense the bone is, how much calcium there is in the bone. We talk about osteoporosis as a silent disease, and that's because bone mineral density can fall, and there's no there's no receptor to detect that. The receptors, the calcium sensing receptor, and all the other mechanisms we have are all very focused on keeping calcium concentration in the blood within a very fine margin. And that sort of helps me look at this from a different angle. And it's always refreshing to look at things from a different angle because, as I say, we focus very much on the bone, whereas the body is very much set up to look at calcium, in the bloodstream. And I suppose leading on from that, you mentioned we're always interested in new avenues of exploration for therapeutics, for medicines and drugs that will help things. And most of the medicines we think about and we explain to patients about as very much sort of in the bone. So bisphosphonates will stick to the surface of the bone and inhibit the action of osteoclasts. Denosumab will switch off the maturation of osteoclasts. Teriparatide, romosozumab will be there promoting osteoblast. It's all very much in the bone, at the surface of the bone. Raj was actually saying to us, look, there's opportunity here to look at a much, much higher level, to look at these calcium sensing receptors, either at the receptors themselves, or even he mentioned the intracellular apparatus that transmit the signals from these receptors. And perhaps in the future, there may be ways of regulating, the thermostat for calcium control at a much, much higher level rather than just sort of at the cell surface. And I often say to people, and I often say this in talks, we only have two levers to pull. We can either push the osteoblast lever or pull back on the osteoclast lever. Maybe if we go on to develop therapeutics based on regulation of calcium metabolism at a much higher level, maybe I'll have to change that and say we've many other levers we can push and pull. And I suppose finally, as you know, I enjoy a bit of history and a bit of philosophy, and Raj and I, even after we stopped recording, had a great chat about the history of medicine and so on. And it was interesting to hear him saying, as editor of the JBMR, one of the the biggest, if not the biggest, journals in the world in terms of bone mineral research and osteoporosis research. And he's commissioned this series where people are going back and looking at older articles that they think are impressive or important or should be remembered. And I must say I'm a big fan of that because there's so much when you look at the past, you can see how people thought in the past, how things were worked out. It's interesting to see blind avenues that were followed that didn't turn out to be useful. And I love reading back over sort of medical history to learn how the people whose work we based on, I don't remember if it was on the podcast or not, or if it was after we finished, we used this expression, standing on the shoulders of giants, which I think was Sir Isaac Newton, and how there's so much we do. I, as a clinician, and certainly you, Richie, as a researcher, were standing on the shoulders of people who worked in the 1990s and the 1970s and the 1950s, and in some cases back hundreds and hundreds of years ago. And it's always interesting to see how their minds worked, how they reported that, and to see what we can learn from that. So I was delighted to see, even though Raj is as a world-renowned researcher and world-renowned clinician and world-renowned patient-facing expert, he actually loves a bit of medical history and a bit of medical philosophy the way that I do. And I thought I really enjoyed that as well.
Yeah, that was nice. I enjoyed that. I guess, that's why he's a leader in the field, because he ticks all of those boxes. He's really helped further bone science, not just through his own research and explaining that to everybody, but also through his decades of service at JBMR. now as editor. It's such a crucial role in our community that we have a good journal with rigorous peer review that tries to pick and collate the best papers for us all to read. It's really, really wonderful, and I'm really glad he's at the helm.
And we're so fortunate, Richie, with Raj and so many other national and international experts to give up their time and come and talk to us on the podcast for some reason. So we're always slightly in awe of the fact that people want to come and talk to us. There must be something good that we're doing.
Yeah. If you're thinking about becoming a guest on the podcast, I think you'll have a good time. It's a fun experience, and hopefully word is getting around, and it does seem to be getting easier and easier to persuade people to come and join us.
Well, that's great. So we look forward to seeing you again, folks, later in 2026. Don't forget, it's the year of the osteoblast, and we hope to see you again soon.
Bye, everyone.
Bye now.